questions, I'll go ahead and start. We're a few minutes ahead of schedule, which is lovely. So I'm going to give the last of the talks related to this edge proposal. And what I'm going to tell you about are tools that exist in mice that don't work very well in other systems that allow you to do really quite non-invasive, very simple modulation of gene function. And this work is being done here at UT, and it's really a collaboration here at two labs. Boris Zemmelman is in the Department of Neuroscience here, and he's just a really fantastic collaborator, and his lab is doing all the actual AAV packaging, and has helped us get this kind of novel, strange strategy that we came up with going, I'll tell you about. And then in my lab, Joel Tripp is a postdoc. He came from Andy Bass's lab. He's got a poster on this stuff as well. He's been here for about 6 months now, just getting started on this project. And then he's working in Boris's lab with Madeline Fletcher-Harrison. And so Madeline is actually doing a lot of virus packaging with us, and while Joel is doing the other aspects of the molecular biology and the animal work. So what I'm going to talk about are just a few kind of broad things. I'm going to tell you what the goal of the project is, why we're bothering to do it, how it fits into the EDGE goals overall. I'm going to introduce you to the AAV, which I know most of you have probably heard of, maybe not all of you. But I'm going to talk a little bit about its biology just because it's relevant to what we're doing. We're going to talk about the directed evolution of AAVs and then our particular strategy and where we are in this process. So Dave mentioned the work that he's doing with the transgenic manipulations, and we see the power there. Obviously knockouts give you the germline manipulation and knock-ins. It's a tremendously useful thing to do, and then once you have the line, you can keep using it, relatively easily with relatively low cost, and obviously that's been really powerful in the mouse world. And you can cross those with tissue-specific lines to alter the timing or location of expression. As Zoe alluded to, that's not the only tool that we want, because one limitation here is the actual generation of them is costly. Unlike the mouse world, where there's a huge number of researchers all generating lines that contribute to the possibilities, we have a much smaller community, even though it's substantial, and so that limits some of the potential use. Zoe talked about doing gene therapy methods by intracranial injection. That's also very powerful and a really logical complement to these transgenic methods. It gives us regional specificity, but it's also a little bit labor intensive and has some significant costs per gene. And it's valuable in that it's targeting specific neuronal populations by the timing and placement of injection, but that can also, sometimes you might want to target the entire brain in a more systematic way, and it's not really a great tool for that. And so the third arm of this is developing a method for intravenous injection. The great thing about this is that when it works, it's simple and inexpensive. So as a user, all you need to do is be able to target the target the vein in the retroorbital sinus, which is harder to say than it is to do. And then the result can be a whole brain manipulation. So this is really much less invasive, and you can then potentially gain specificity through promoters and enhancers, although those have their own challenges in terms of cell-specific populations. But we think this is going to be a really nice logical complement to these other approaches. And so I'm going to tell you a little bit about those. So what is the adeno-associated virus? It's a single-stranded DNA virus, and it's really pretty simple. There are just two open reading frames, this rep and cap, and then they're flanked by these terminal repeat sequences, these ITRs, inverted terminal repeats. And those inverted terminal repeats, so the rep proteins, I say here that there's sort of two genes because there are actually multiple proteins that are encoded from these sequences. But the rep gene is involved in regulating the expression of the cap gene. The cap gene actually makes a series of proteins that form the capsid coat of the outer part of the virus. And so while the rep helps regulate the expression of the capsid, it doesn't, the AAV by itself isn't actually replication competent. That's one of the reasons it's so safe, and that's a BSL-1 level agent, is that it's not replication competent by its very nature. It needs another virus, like an adenovirus, to help it replicate. And so that's why it's called an adeno-associated virus, because in the wild you find it in places where you also find adenovirus. So that safety then is really a powerful aspect of it. And then also the fact that it tends to be episomal, that it doesn't get integrated into the DNA, also contributes to its safety. And then it's long lasting too. So in order to harness the use of this virus as a gene therapy vector, and this is really, the most, you guys know this, but by and large, this is really the most common sort of gene therapy vector. But in order to make it, they do a couple tricks. And so these ITRs are what tell that chunk of DNA that it can get taken up. It's what gets recognized by the capsid proteins as the viral particles assembling. And so the ITRs are putting whatever is in between, if it's not too big, inside the viral particle. And so you can play a trick on the virus and swap out these two open reading frames with a gene of interest and a regulator, flanked by these ITRs, and then the result is going to be that that piece of DNA will get taken up by the viral particle, and that's how we make a gene therapy vector. But then to do that, the rep and cap have to be in the same cell at the same time, and you can do that by putting them on a separate plasmid and transfecting them. Well, as I mentioned, it also needs other proteins from adenovirus, and so you can put those necessary proteins on another plasmid, a third plasmid, co-transfect all three, and trick the cells into making these viral particles that are so useful. Okay. So there's a lot of natural variation in the function of the AAV. And when people describe AAVs, they're usually, you'll see a pair of numbers separated by a slash. It's usually AAV2, which refers to the backbone, which really just refers to the ITRs from one particular strain of the AAV. And then the second number refers to the strain from which the capsid is taken, which is what assembles the outer coat. And then those different strains, the different capsids, might be better at infecting some kind of cells than others. They have a tropism. Well, they have a tropism because there are differences in those capsids, those coat proteins. And we don't have to stop at the natural variation that you can find in a capsid. And so one thing that people have done quite a lot is they realize there's a certain point in the capsid sequence where you can insert a peptide that's 7 amino acids long, and it's called the heparin binding site. It doesn't really hurt the structure of the capsid, and that's because that little span of amino acids sits out on the outer part of the coat and doesn't interfere with the coat assembly at all. So if you insert a little piece in that part, by adding that piece in, those amino acids are going to have their own distribution of charge, their own shape. All those things are going to influence how they interact with other proteins. And so if you make a random library of 7 amino acids, that library can be huge, right? So 21 to the 7th is quite a large number. And so then you can screen that library, and it's kind of a random roll of the dice where any given peptide will change the function of the capsid in a useful way. But if you have a powerful way of screening it, then you can select for viral particles that have useful properties. And in order to do that, you have to do one little trick, which is you have to not have a gene of interest that you're trying to drive expression of, because this is a development tool, not the actual implementation of the tool that's been developed. And to do that, you take the capsid and put it back between the ITRs, because what you want is whatever weird 7 amino acid sequence is in that capsid, you want to also be in the DNA that's inside that virus. Then you can select for the viruses that have the interesting properties, sequence the capsid sequence, and then once you've isolated something that does what you want it to do, that interacts the way you want it to with other proteins, then you're off to the races and can do some other things. And so this has been done to develop AAVs that have that are really good at getting inside the retina, that are good at getting across the blood-brain barrier or evading the immune system. These are all kinds of things you can select for. And one of the more dramatic things that was done by Deverman et al. Out of Viviana Granaro's lab, and what they did was to realize that, it had been published that certain serotypes of AAV could get past the blood-brain barrier and infect neurons. They didn't do it very well, but they did do it. And so they reasoned that they could take this directed evolution approach, generate a random library of peptides, and then find ones that actually were effective at doing that. And so they did that, and they developed a strategy they call CREATE. And so here's what the CREATE strategy looks like. So this is the payload that's inside the virus. I'm not showing the inverted terminal repeats here, but they're there. And they have a gene that, a reporter gene, M-cherry, being expressed by a strong general promoter. And then they have the capsid gene. And then this rainbow indicates that there's a whole, that it's not just really one sequence, that each plasmid has a unique 7 amino acid sequence there, or a unique 21 base pair sequence. And then this replicator is actually a promoter that's driving the expression of the capsid, just when you're making it. So they call this the cap in cis lock. So it's in cis because the cap is in the same piece of DNA with the rest of the stuff, including these Cre-sensitive lock sites. And then they use these kind of interesting sites that when you're exposed to Cre, recombine irreversibly. And so they take what would normally be the polyadenylation signal, they flip it over, and now it faces that way. And so if Cre is present, wherever this DNA is present, now they could take primers that were both originally facing that way and so wouldn't work, now facing each other, then they can amplify the intervening sequence, which includes this little piece here. They can try and recover the capsid sequences that work well. So that's what they did, and after a couple rounds of selection, they found that the natural ability of AAV to, when injected into the retroorbital sinus vein, could not only, this is what the wild type AAV9 serotype looked like, and so it showed some ability to get into the brain, really go to the brain stem. And then they took a line of mice that expressed Cre, and then by expressing Cre in the brain, they could select for the viruses that were able to get past the blood-brain barrier, invade the immune system, and infect cells. And when they did that just with a couple rounds of selection, this is one of the things that they isolated that had really broad infection ability. And so just from a single injection, then you can then potentially infect the entire brain. And so that's obviously really powerful and really exciting. And you can imagine that if we could do this with voles or with other species, like, we could do a lot of interesting things. We could have activity-dependent promoters. We could be manipulating the particular genes. We could, maybe put, maybe this is a tool by which we put guide RNAs a lot of places. Lots of possibilities. The trouble is that when folks have used these, the first generation capsid that they developed didn't even work with other strains of mice, unfortunately. It didn't generalize. The selection regime was very specific to the mice they used. Didn't work on rats. Zoe tried it with voles. It didn't work very well. So this is a mouse that they injected intravenously. And so these little dots here are labeled cells. Voles, you see a few, but not very many. So it's kind of similar to the wild type virus in that it does it a little bit, but the selection procedure doesn't seem to really help here. A colleague of mine, my collaborator, working on singing mice, also tried this with the singing mice. I told him, don't bother. Zoe did it. Didn't work on mice. Or it doesn't work on voles. It also doesn't work on singing mice. They see a few scattered cells in a similar way. And it does seem to be something about the ability not to infect neurons, but to either evade the immune system or get past the blood-brain barrier, because when Zoe in her lab injected it ICV, she did get good expression and good infection, and the injections work well. You can see if your injections work by injecting a small amount of ink. And so if you do that, the skin turns all blue, the brain will turn blue. And so it's not hard to learn how to do this mechanically. This is all working. But for some reason, this particular capsid protein is not working. Okay. So What we want to do then is use a similar kind of approach, but find something that'll work for non-model organisms. And there are a couple strategies, and there are a couple ways we thought we could tweak this general idea to do something that might be more generally useful for people working in non-model systems. Well, one thing is that we want to find something that works across mirrored rodents. And mirrored rodents is a large group, and so the voles are here, and the arbiculines. This is a recent phylogeny from Steve Steppen. And then the lab mice are murines here in black. And so they're part of this very large assemblage of rodents called the Muridae. And so Muridae is something like 40% of all mammals. It's an enormous group. And so when you think about the possibilities for non-model systems beyond voles, it's really, really quite cool. So we can address that, I think, simply by doing the selection of AAV particles in parallel across multiple species. So selecting not only in lab mice, which we'll continue to do, but also in prairie voles, and we're adding also singing mice, another group that my lab works on. And singing mice are also in Chrysalidae, but if something works in all three of those species by design, by our selection regime, then it should work across all murids at least. And so the hope is that we'll have something that Peromyscus people can use, and lots of folks, gerbil people, hamster people can use, all kinds of people would be able to use this then. So that's easily implemented just by a change in selection. There are some other complications though, because the strategy that they developed really relied on having a Cree line or some Cree expressing animal that you could then use to drive the, use to recognize when stuff got into those cells. And that's a problem for non-model organisms because you have to get the Cree there somehow. Now we could go in earlier and do a separate virus injection and try and get it in lots of brain regions. But as we're thinking about it, I thought, well, wouldn't it be a lot more elegant if we could just eliminate that aspect of it and come up with some new strategy? Another thing is that traditionally when people have done these selection experiments on AAV, they've PCR amplified the pieces like I showed you, but they've just sort of Sanger sequenced a handful of clones from those. And so they really haven't taken advantage, with the exception of one example I can think of. They generally haven't taken advantage of really the power of current sequencing methods. And so we want to do that, and so we can exhaustively characterize the library. So we came up with a strategy, which we've just decided, as I was writing this talk, to call DICE. Partly because it's a bit of a gamble, taking advantage of the natural gambling nature of evolutionary phenomena. And I mentioned to Joel that if it didn't work, we could call it no dice. And then Joel said that it would make it a dicey strategy for his career. So, so this is what we're doing. It's called DICE because we're calling it directed evolution by inverted capsid expression. And so this is the main construct that we made that implements this strategy. So we have, again, the capsid gene sequences in cis with the reporter sequence, and it's going to get taken up by the viral vector, by the capsid that's produced by it. But what's weird here, notice, is that they're facing opposite directions. And the reason that's useful is what that means is when this plasmid is in a kidney cell that we're using in culture to generate the virus, the rep promoter should be good at driving capsid expression just like normal. The HSIN will give us a little M-cherry, hopefully not too much. But then when we take that packaged viral particle and put it into the nervous system, this HSIN will drive M-cherry really strongly, but we shouldn't really be getting any capsid expression in the brain. And instead, all of this stuff here will show up in the 3' UTR. And that's really powerful because then we can use sequencing to figure out just the pieces that make it into the brain. And then we can sequence those and instead of sampling 10 clones, we can sample literally millions of sequences and really get quantitative estimates across tissues, across species, and as the selection experiment unfolds to see what happens. So that's the general structure. And so we'll take this plasmid, and we've got the same library of 7 amino acid insertions there. We'll make a randomized capsid library that'll have a bunch of viral particles. We'll inject the AAV intravenously into the retroorbital sinus. We'll wait 2 weeks. And then we'll do is we'll go to several tissues. We'll go to the cortex, we'll go to the hypothalamus, we'll go to the cerebellum. We'll harvest the RNA from them. And we'll do that for several males, several females, in prairie voles and singing mice and lab mice all together. So we'll have then we'll have 9 different tissues, those 3 tissues from 3 species, plus multiple animals of each sex. So it adds up, and we'll have a lot of different tissues. And so we have kind of an elaborate sequencing strategy that I think will help us with that. But then what we're going to do is, we've got these primers that then amplify this piece. But what's unusual here is that we're putting on, we're taking kind of a trick from metagenomics or environmental genomics, and we're adding on sequencing adapters to the end of those primers. And so that when we sequence those pieces now, when we PCR amplify them, we can take one aliquot and then send it off for sequencing. And they'll be barcoded, and so each of those individual samples from each of those individuals, each of those tissues, we're going to know how reliably these things work. We're going to know whether they're things that are specific to certain tissues or certain species, and all that information will be preserved. But then we'll take all those, from all those tissues and all those species, all those individuals, and we'll pool them. And then we can just cut them at the restriction sites, which are still there, and then reclone them in and make a new library, and then do the selection again. And the hope is that after a few rounds of selection, we'll have something that now we're screening a much, much larger population of viruses. We're doing it in a way that samples a much broader range of genetic variation, of lineage variation, and so we're hopeful that this strategy will give us something we can use that will produce a very broadly useful construct. So far, we've assembled the DICE plasmid, the main plasmid I talked to you about, and Boris and Madeline have also assembled the various helper plasmids, which are a little more complicated than the normal AAV production plasmids for various reasons. Generated randomized capsid inserts and sequenced them and confirmed that they retain some diversity. And so we're currently testing the ability of the protocol to generate viable viruses, and so we should know in the next week or two what's going on there. And then the retroorbital injection worked as expected. So what we're doing now is the next 2 major steps before we actually really get to do this. We're cloning this library into the DICE plasmid and confirming the diversity by sequencing and confirming our sequencing strategy works. And then we have to titrate the concentration of these plasmids to find the right combination that still gives us a lot of virus, but maximizes the likelihood that the plasmid that's in the cell is the same one that gets taken up by the capsid. Now that's a strange thing. Normally if you're making a virus and you're transfecting all these plasmids, you might get multiple capsids, plasmids going to the same cell, right? And then you wouldn't care because they're all the same. But in this case, we do care. What we'd like, ideally, is in any given cell, there's only one plasmid that has a capsid. And that capsid then would be the only thing that could be taken up by a viral particle. And so, and then the viral particle will necessarily have been produced by that capsid sequence. And so there'll be a one-to-one relationship. Anyway, so figuring out that balance is kind of the last, I think, technical hurdle that we have. And once we do those 2 things, which we're working on now, we'll begin selection. And then the last thing we have to do is cross our fingers. But no, I'm very hopeful about this. I think it's a really exciting strategy. And if it doesn't work, we can go back to the create strategy if we need to. But we decided this was kind of powerful enough that if it worked, it was worth trying. So that's it. And we can take some questions.
That was really cool, Steve. Thanks for doing this. The non-invasive approach is really appealing. I have, I think, 2 questions. The first one's sort of basic. Could you back up maybe 2 or 3 slides? It was about the repeated approach you're doing, where you take the 7 amino acids and then put them back in the hopper, basically.
Uh-huh.
Does that mean, if I understand this correctly, that, you know, the first round is gonna have 7 and then 14, 21? Is it?
Oh, no, it does not mean that. Thank you for asking. I'll show you another. This is just something that a similar slide, but focusing really on the sequencing strategy. So these 21 base pairs, initially the library is 21 base pairs at random. And then after we go through a round of selection, we're trying to enrich for that combination of 7 amino acids that gets past the blood-brain barrier, gets past the immune system, preserves the ability to get into the neurons. And so when we so those that do will then get into brain, get into neurons, the HSIN promoter will drive the expression, and then that'll show up as messenger RNA. We'll amplify that, sequence it, and then that stuff that we amplify then is going to be a different library than the library we start with. Because now it's going to be enriched for things that are able to do that. But we're not assuming that everything that gets there, you know, like there's not going to be a one-to-one relationship between the plasmids and the cell and the capsid. So there's going to be like some of the things just by chance are going to do well in the brain. So we're going to keep doing that repeatedly until like after multiple rounds of selection like that, then we'll lapse it. So we generate a new library of sequences with recloning the plasmid, and now it's enriched for things that work.
Oh, that makes much more sense.
So then we're just repeating that enrichment, but it's still always the same length, the same size.
And my second question was, and this is probably not an issue, but do they have any concerns that these 7 amino acids aren't so much bypassing the blood-brain barrier as impairing it?
Yeah, no, that's a good question. I think, so Deverman now has his own lab, and he's been looking at the exact mechanism by which it's bypassed. And so there's a specific receptor that's interacting with. And I don't know off the top of my head what kinds of things they've done to look at the integrity of the blood-brain barrier. But that's actually a good point that I should check and make sure that that's not a possible explanation. But you know, the animals are viable and look great, so presumably that's not the case. But given this other work on the specific receptor type, I would imagine they've looked at the integrity of the blood-brain barrier. But that's a good point. Can you speak to that directly?
So what doesn't happen is a massive inflammatory change, which is good. But that was actually a significant concern. There is actually increased permeability to both peptide as well as non-peptide compounds. It's not enough that there seems to be huge changes. There's a couple of people have looked just transcriptionally, both within the endothelium as well as in neurons. And so there are definitely changes, but they don't seem to have global effects. And whether or not at the end of the day, it's something that you could, these are people that are trying to think about this from a clinical perspective. So there's enough that people are worried about it in that context, but from an animal model system, it doesn't seem to have global problems.
That's really interesting. You know, it could be that at the end of this, we're going to have not 1 or 2 sequences. I expect we're going to have a lot of sequences, right? And it might be worth thinking about when we're doing, you know, when we're exploring the efficacy and the consequences of using this capsid, that might be something we look at, and we might then manually compare multiple serotypes to see, to look for something that sort of minimizes some of those issues. That's a great question.
So you're not doing any mutagenesis right around, is that right?
No, we thought about it, and you know, one possibility is that we could start with the best capsid from the Garnaru group and randomly choose 2 amino acids and then randomize 2 at a time. So we're kind of going 2 steps away from that, kind of searching that space. And I think that would be another good way to do it. But it was actually easier and cheaper to exhaustively, like, you know, it's very easy to specify a random 7 amino acids or random 21 bases. To specify, you know, choosing 2 and all the combinations of those, it's 21 different oligos that you have to pool together to make a library out of. So that was the basis. It was just a little simpler. There are good reasons to do either, we thought. Like, it's possible that the sequence they have is in the vicinity of something that would work more generally, but we thought probably be a little safer if we started with a larger library, given that we're gonna screen it on multiple species.
So Steve, I think all these technologies are incredibly exciting, but I think it's also worth a quick, just, what are the limitations of this system if it does work?
Yeah, right, so, I mean, the limitations of this, really, I think, is that, you know, it's by its design and by its nature, it's only really good for whole brain interventions, right? And so you could use it in combination with something else potentially, and then, you know, the other thing is the size of the packaging again, like, probably, on its own, this wouldn't be good for, you know, delivering a dead Cas9 fuse to the activator, right? So, all the things that you talked about that are limits of AAV will be limits of this approach. In addition, the trade-off between non-invasiveness and ease of use and local action, you know, like, yeah, that's something that this method is not the right method for that. If you're interested in really specifically targeting a specific population by injection. But, you know, if these directed evolution strategies could be really super useful, like one thing that would be amazing for this would be, you know, if something like this works, we could screen promoters on a really big scale and do all kinds of things and maybe get around some of the specificity challenges that we're currently addressing with injections and get around those by more systematic screening of sequences. Like that would be really
I also want to just mention quantity of virus required.
Oh yeah, that's a good point.
So this is not inexpensive. You're injecting 100 to 200 microliters of virus at titers you would use in the brain intravenously. So you need huge amounts of virus if you're going to do a cohort of animals.
That's a good point, Zoe. Thank you.
No trainees. Are you trained? No.
Sorry guys, trainees.
No.
Yeah.
Okay. Here. Okay.
Okay, this might be a naive question, but so by design, this is specific for the AAV. Are there aspects of the directed evolution that could be adaptable for like the lentivirus or the HSV?
Yeah, no, I mean, I think, you know, the strategy that this inverted capsid in the UTR is a very general strategy. Like it could work, you know, you could do that for code proteins for lentivirus, for other things too. I haven't thought about those in detail. You might also be interested, like you could also use this exact library to screen for things that just worked in a fish, right? Like you can do an ICV injection. And we don't have gene therapy vectors for lots of taxa outside of mammals. So, yeah, so I think it's general both in the context, both conceptually in terms of the virus that you would use, but also taxonomically.
So to follow up what Zoe was just challenging you on and what you said, can you elaborate a little bit, assuming your gamble is going to be successful, which of course it will be, how you would go about getting cell type specificity?
Yeah, so, well, so it probably not that many people in this room other than Deb and Zoe and I like follow the nitty-gritty molecular stuff that much, but this strategy owes to a couple different things. One, this idea of putting in the 3' UTR is derivative of a massively parallel reporter assay called StarSeq. And so there, the idea is you put a regulatory element in the 3' UTR, put a whole library of elements there, or an entire genome there, and then you put them into the animal, and then you see, and then you use the 3' UTR sequences to figure out which ones are acting as regulatory sequences, right? We could do that with this. And so if we had a library that had M-Cherry and then had a library of enhancer sequences, put that through the whole brain, do single cell sequencing, you could find out, oh, well, here are the sequences that drive expression in dopaminergic cells, here are the sequences that drive expression in vasopressinergic cells. It's more than doable. I think it's actually, I mean, that's honestly my interest is in regulatory evolution, and this for me was a way to try to develop skills that be related to trying to ask some of these hard regulatory questions.
One
where you get cell type specificities is use this in conjunction with like Cree animals. So like if you had some gene Cree,
Yeah,
right, then you can inject and express in the whole brain, just in those, that set
Yeah.
of cells.
oh, for sure, for sure.
But the limitation of that is like, what percentage of neurons in the mouse study that, where they first reported it, what percentage of neurons actually was transfected?
I don't know the number off the top of my head, but it's really abundant. And it's abundant enough to be, do you know off the top of your head, Zach?
Yeah, it was close to 90%, but it very much depended upon the titer, and you need to get up to like 10 to the 13th and inject almost 200 microliters to get to that point.
90%'s a lot.
And it is somewhat biased, so different cell types, you can look at different types of neurons, and there's some variation in how well this works.
So, I'm not seeing in the construct anything that says that this will affect only neurons. So it could also be.
Oh, yeah, no, but the selection regime is driven, so because the selection depends on expression, and the expression is driven by a neuron-specific promoter, or HSA, we're only selecting for things. Now, that doesn't mean that what we pull out might not also express in glial, right? And that's something we could, so once we develop some things, we'll take a handful of different constructs, package them, and then characterize them. And the characterization will look for, bad effects, and we'll look at the specificity of expression, and we'll look for tropism across different regions of the brain and stuff like that. But I think the tropism will be well addressed by the sequencing data, because we'll have a good sense for any one of these constructs, the degree to which they're specific to a region or a species, at least for the regions we look at.
So the peripheral nervous system is also affected?
Yes. Yeah, right. We're not directly selecting that, but yeah. Questions not expected. Exciting though. Well, it is just now 11:31, so our lunch should be waiting for us. We have an hour and a half, that's right out where the tables were. And say hello to folks at the sculptors, while you're out there.